In this lecture, we are going to discuss about microscopy. These are the objectives which have to be covered during this lecture, as well as the practical, which will be held later. Microscopy is the science of observing structures that are too small to see with the naked or unaided eye. In order to visualize structures that are too small to see, we use the instrument we call the microscope. The microscope has the ability to enlarge objects so that we can see them. And this enlargement of the objects is called magnification. Let us look at the history of microscopes and microscopic world in brief. In 1650, Anton van Leeuwenhoek invented the first light microscope, and he is known as the father of microscopy. Several years later, Robert Hooke first described a cell. He looked at a piece of cork through a microscope and coined the term cell. In the 1800s, a lot of inventions were born. First, the compound light microscope. Then Brown discovered the nucleus. Then the cell theory was introduced by Sheldon and Shawn. Cell division was discovered by using the microscope. And the description of the 4 basic tissues happened. In 1932, the microscopic world took a big leap when Knoll and Ruschka invented the electron microscope. In 1986, they won the Nobel Prize in Physics for the invention of the electron microscope. This opened the gateway to the study of the ultrastructure of cells and other structures. Now let us look at the different types of microscopes. Optical or light microscopes are the ones that were first developed and discovered. Optical light microscopes use light to create an image of the object that you are viewing. These microscopes have the ability to magnify the object 500 to 1000 times. Then there are the electron microscopes. They use a beam of electrons to create an image of the object. They can achieve very high magnifications, about 100,000 times. And you get the chance of observing the ultrastructural features. Now let us look at light microscopes. Light microscopes are commonly available. If you go to a hospital or a laboratory or a research facility, the most commonly available microscope is the light microscope. Light microscopes may use sunlight as the source of light or an internal light source which is built into the microscope. Given below are pictures of the 2 types of microscopes that are available in our microscopy laboratory. Now let us look at the basic concept or the basic principle of light microscopy. A beam of light is passed through the biological sample or a tissue specimen, and the image is magnified and projected into the retina of our eye, and that is, we call this as a visual medium. In this diagram below, you can see that the image of the tissue sample is created in the retina. And the microscopes magnifies and enhances and helps us to see the structure let us look at the light microscopes in more detail. Light microscopes which are available now are called compound light microscopes because they magnify the object twice. They have two lens systems. One lens system is called the eyepiece lens system and it is close to the eye. The other lens system is called the objective lens system and it is close to the object. This diagram shows you how the object is enlarged twice or magnified twice so that you will be able to see it properly. You can see that two biconvex lenses are used here. Okay, and it increases the size of the object. Now let us look at the parts of the compound light microscope. This microscope that you see here uses sunlight as the light source. Let's go from top to bottom. On top, you can see the eyepiece lens. That is the point where you have to keep your eye. Then you can see a knob called the close focus, which moves the eyepiece lens part up and down. Then below that, you can see another knob, which is called the fine focus, which again moves it, but very small amounts. Then below that, you see the round area, which is called the nose piece, and attached to the nose piece are the objective lenses. In this microscope, there are 3 objective lenses. Then below that, you see the stage with the clips to hold the histology slide. Below the stage, you see the condenser, which directs the light onto the stage. Then below that, you can see the mirror, which helps you to focus the sunlight onto the microscope. You must always hold microscopes with both your hands by keeping one under the stage of the microscope and holding on to the part behind so that you don't damage the microscope. Here you see a different or binocular microscope which has 2 eyepiece lenses. This is slightly more complex and you can get a better image from this. Below the eyepiece lenses, you can see the nose piece and the objective lenses, which there are 4. You can see the stage, and on the stage there is the clips which hold the slide, and you also have knobs that can control the slide and move the stage. Below the stage, again the condenser, and below that, instead of the mirror, you have the light source which is inbuilt. So you have to plug the computer into a power source before you can use it. And you can adjust the intensity of the light. On the side, you can see the focus knobs. They are both on the same knob. The inner circle is the close focus knob. When you try to move it, you can feel it that it moves a lot, moves slowly or with difficulty, while the fine focus knob moves pretty quickly and with ease. Now let us look at the different objective lenses. There are 4 different magnifications. The lowest magnification is 4, then 10, then 40, and then 100. You can see bands of color around the objective lenses. So basically they are color coded. 4, 10, 40, and 100. On the 100 objective lens, you can see a small word written called oil. That means the 100 lens is used for what we called oil immersion. The eyepiece lens is commonly has a power or a magnification power of 10. How do you calculate magnification? So magnification means how much the object is enlarged. In order to calculate the magnification, you take the power of the objective lens and the power of the eyepiece, and then you get the magnification. So generally you write it, if you're writing about a slide, you say 10 to the power 40. So you know that that slide is focused in what we call high power. 40 or 100 is considered as high power, while 4 and 10 are considered as low power. Okay, so firstly we write the power of the eyepiece lens as 10, then we put the X, and then we write 40. So if you're asked to write the magnification of a slide, that is the way you do it. You say 10 into 40 equals 400. Or other way around also you can write. Now this chart shows you the units of measurement. I want you to understand the different structures and their relative sizes. Now the smallest amount that you can measure with a ruler would be 1 millimeter. Okay, so if you look at the pituitary gland, which is a gland in the base of your brain, it is about 1 centimeter in diameter. Then if you look at the intestinal villi, that would be about 1 millimeter in height. The largest cell in the body is a oocyte, and it is about 100 microns. You may be able to visualize this with the unaided eye. Then a red blood cell is about 10 microns, while a bacterium is 1 micron, and the virus is about 0.1 microns. Then of course you come down to the other molecules which are smaller, up to the hydrogen atom, which is the smallest molecule. So understand the sizes of different structures in biology that you can see. So with light microscopy, you can observe a bacterium, but to see a virus, you need an electron microscopy. As I said, a mature oocyte may be just visible to the naked eye because it's about 0.1 millimeter in size. All other human cells are microscopic. A liver cell is about 50 micrometers, and any structure which is less than 500 nanometers requires an electron microscope for viewing. Now let us talk about the concept we call resolution. Resolution is the amount of detail that you can see, or the sharpness or clarity of an image. There's actually no point in just enlarging the image or enlarging the object. You have to make a sharp or clear image. As you can see below, the A image is clear. It is sharp. The edges are sharp and you can see more details than the B image, even though both these images are of the same size. So resolution means making an image clear, clarity of an image. Objective lenses magnifies as well as enhances the resolution of the image. So the eyepiece lens and the objective lens together gives us a magnified, enhanced image with high resolution so that we can observe the structures. I told you in magnification of 100, if the objective lens is 100, we call it oil immersion, and we will focus it on to the 40 objective lens and then turn it on to the 100 one, and you will see that it does not have a good resolution. So in order to see in oil immersion or 100 magnification lens, you have to drop a tiny bit of oil onto the slide. So in the picture below, you can see when the oil is not there, it is not clear. When you add a bit of oil into it, the resolution improves and you can see it. So what the oil does is to increase the resolution. Now, in order to visualize tissues, you have to do what is called tissue processing. In order to visualize the microscopic structure of human tissues, the fresh tissues have to be processed. Tissues are infused with paraffin wax and then sliced into thin sections. Then these sections are laid onto glass slides and stained with color. Then only you can see proper structure through the light microscope. Now let us look at the two colors or dyes as they are called, which are used to stain histological slides. The most commonly used dyes are hematoxylin and eosin, and we write it as H and E. Hematoxylin is a basic dye which stains acidic structures in purple. Eosin is an acidic dye which stains basic structures pink. The acidic structures you can see in a cell are the nucleic acid, glycosaminoglycans, and acid glycoproteins. The basic structures are mitochondria, cytoplasmic granules, and collagen fibers. Here you see a slide stained with hematoxylin and eosin. The structures stained with hematoxylin are the nuclei. You can see the granular appearance of the nucleus. The chromatin in the nucleus is stained with hematoxylin. Then the structures which are stained with eosin are the pink ones. That is the cytoplasm as well as the collagen fibers. Now let us talk a little bit about other microscopes. First, let's look at a digital microscope. A digital microscope is similar to a light microscope. However, it is attached to a camera and to a computer, and the image that you focus on will appear on the computer screen. Now let us look at a different type of microscopy which we called fluorescence microscopy. In this type, the tissue sections is irradiated with a specific band of light, and then the fluorescence which is emitted by the cells or structures is detected. Fluorescence dyes are used to stain specific structures. For example, a stain called DAPI is used to stain the nucleic acids of a nucleus blue. Here you see some cell scrapings from the cheek which have been stained with DAPI, and you can see the nuclei colored in blue. So this is a fluorescence micrograph. The fluorescence microscopy can be taken into another level by using what is called immunofluorescence. In this technique, we use antibodies that bind to specific structures, and then these antibodies are tagged with fluorescent dyes, and that can be seen through the microscope. In the diagram below, you see a certain protein shown in blue diamond, and that antibody shown in pink attaches to this particular protein, and that antibody is tagged with a fluorescence dye. So when you look at this through a fluorescence microscope, you know where this particular protein is. These are some fluorescence micrographs which have been taken of a breast cancer tissue. Certain proteins called vimentin, CK19, and the DAPI is stained in red, green, and blue, and you can see the fluorescence or how it looks under a fluorescent microscope. This is a picture showing you the fluorescence microscope. This is again can be a digital microscope where it's attached to a computer, and you use different shields to irradiate the specimen with different colored lights. Next, we look at what is called phase contrast microscopy or dark ground microscopy. This is used to visualize living cells without preparation or staining. The background light is changed in order to visualize transparent specimens. This can be used as a time-lapse microscopy videos to observe dividing cells or cell movements. It can also be used to observe organisms like bacteria or paramecium. Now let us look at electron microscopy. Electron microscopes, as I said earlier, uses a beam of electrons to create an image. It can achieve very high magnifications, more than 100,000 times, and you can observe the ultrastructural features. There are two types of electron microscopes mainly, the TEM or the transmission electron microscope, or the SEM, scanning electron microscope. You do not need to know details of these microscopes, just to remember that they have high magnifications and what kind of images they produce. So the transmission electron microscope, you can have a magnification of up to 400,000. These are the kind of images that you will get from a transmission electron microscope. These are called transmission electron micrographs. The tissues have been prepared using a resin medium, sliced, and then you acquire these images. So you can see a cell with a nucleus and mitochondria, and then another micrograph of a mitochondria on the right, and again two cells. Then looking at the scanning electron microscope, you can look at the surfaces of different structures or cells. Here you can see blood cells. The top picture shows you red blood cell, platelet, and a lymphocyte. Then below you can see macrophages and also cilia on the respiratory epithelium. Then there's a special type of microscope called laser scanning confocal microscope, which is used to increase the resolution of a small selected area. It has the ability to create images at different depths of a specimen. This is a highly specialized microscope which is used for research. Now there may be other microscopes, many other types, but these are the commonly used ones that you need to know a little bit about. Now just let us revise this. We just look at a few pictures and you should be able to identify from which type of microscope these particular pictures have been taken. So this is a light microscopic view, H&E section. This is a phase contrast where you can see a dividing cell. This is a scanning electron micrograph where you can see red blood cells. This is a transmission electron micrograph of a cell with a large nucleus and several mitochondria. And this is a fluorescence micrograph.